Hi, I'm Ilaria Iacobucci, a staff scientist in Dr. Charles Mulligan Lab at St. Jude Children's Research Hospital. Our work is focused on classification of acute lymphoblastic leukemia across age in children, in older children, and in adults. Our work is based on identifying genetic alterations that drive leukemogenesis, and that can be used for stratifying patients according to their own risk. Historically, classification of acute lymphoblastic leukemia has relied on conventional karyotyping, fluorescence in-situ hybridization, and molecular targeted assay for the identification of gross numerical aberrations such as aneuploidies, structural rearrangements such as translocations, and therefore specific non-gene fusions. However, according to these approaches, around 30% of cases were unclassifiable and simply defined as P others. With the advent of next generation sequencing, several investigators around the world were able to dissect this group, identifying different genetic alterations specific of new subtypes. These were not classifiable before because they were characterized by cryptic rearrangements, so not identifiable by conventional approaches, or they were heterogeneous, so involving different partner genes to a single gene, or they were phenocopying established subtypes, so similar gene expression profiles but different founding alteration. By using sequencing approaches, different alterations were identified, and the number of acute lymphoblastic leukemia subtypes has been successfully increased, with the current over 20 different subtypes characterized by a constellation of somatic mutations, rearrangements that drive leukemogenesis, and by different cooperating mutations that are important for full manifestation of leukemia. These different subtypes converge on distinct expression profiles, and by this approach, so by gene expression profile, we can identify specifically different acute lymphoblastic leukemia subtypes. So which are these subtypes? By gene expression and by sequencing approaches, we can identify clusters of well-known established chromosomal altered subtypes such as aneuploidies, or we can identify a group of transcription factors altered subtypes. These include the well-known established subtypes as determined by conventional karyotyping, but also new subtypes that are driven by rearrangements of specific transcription factors. For example, TNF384 gene, rearrangements of NATM1, rearrangements of ATP6 RUNX1, which is well-known defining lesions, but also a different subgroup defined ATP6 RUNX1-like that has a similar expression profile to ATP6 RUNX1 but lacks that fusion. Additional novel subgroups involve transcription factors that are not affected by rearrangement, but they can have a single point mutation that is responsible for disease initiation. And this is a very novel findings that was only identified a few years ago, where we identify a cluster of patients that were characterized by one specific point mutation at the amino acid 80 in the gene called PAX5, which is very important for B-cell differentiation. This single alteration has been demonstrated by functional experimental models that is important for driving leukemogenesis. It does not occur by itself, but is often occurring with additional alterations, mostly in signaling pathways or in tumor suppressor genes, demonstrating that different alterations can contribute to leukemogenesis. A novel subtype is that defined by another single event characterized by one point mutation, this time in another transcription factor important for B-cell differentiation, which is IKZF1 or Icarus. We know very well that gene because many years ago several groups discovered the deletions in these genes that characterize over 50% of high-risk leukemia subtypes. Here there is a very small subgroups, less than 1% of acute lymphoblastic leukemia, but with a very defined transcription factor. And this group is characterized by the mutation NY59Y in DNA binding domain of IKZF1, which is important for functional consequences in this transcription factor. So by the use of modern technologies such as the widely gene expression profile, exome sequencing, in some cases whole genome sequencing, we can classify acute lymphoblastic leukemia patients according to their specific genetic alterations, gene expression profile, and these alterations are very important not only for risk stratify these patients, but also for guiding novel therapeutic approaches. For example, let's think to the groups of ALL that are driven by tyrosine kinase alterations such as BCR-ABL positive ALL or PH-like ALL. In the case of PH-like, we have over 60 different rearrangements that drive signaling pathways such as ABL pathway, JAK-STAT kinase. So here we can have multiple alterations by the same pathway that is affected, that they can be inhibited by specific tyrosine kinase inhibitors, and these are widely used in clinical trials nowadays. In the case of other novel subtypes, we could suggest novel therapeutic approaches. For example, the focal adhesion inhibitors for IKZF1 alteration, or FLT3 inhibitors for mutations with the rearrangements of TNF384 that are characterized by overexpression of the FLT3 gene. And we know that there are several FLT3 inhibitors that can be effective in this subtype. So implementation of sequencing approaches into the clinical workup of acute lymphoblastic leukemia may definitely be helpful in classifying those patients and in helping in guiding the best therapeutic approach.